Hello, everyone. Welcome to Creative Biolabs webinar session. And the Creative Biolabs is highly specialized in advanced antibody biochemistry and engineering. We offer our customers with comprehensive one-stop shop for antibody-drug conjugate research and development. We are experienced in performing several conjugation strategies for antibody-drug conjugate developments, especially in lysine or cysteine-based conjugation, enzyme-mediated conjugation, and natural amino acids UAA-based conjugation, interfusion technology-based conjugation, and UV photo cross-linking. We also offer a variety of antibody-drug conjugate products to serve the course development and evaluation. The theme of today's webinar is novel design strategies for effective next-generation nuclear localization signals, NLS therapeutics. In fact, research on the implementation of NLS therapeutics has been a topic for intense interest since the core principles governing nuclear transport were awarded to Nobel Prize in 1999. And in this technical session, Creative Biolabs has invited Dr. Leyton to describe the needs to develop a more precise method in investigating nuclear transport dynamics of NLS therapeutics. Moreover, Dr. Leyton will also present a de novo approach for generating novel NLS with a typical zero net charge, as well as evaluating various strategies to determine the worst for integration into NLS therapeutic developments. Next slide, please. And our speaker of the day, Dr. Leyton. He's an associate professor at the Department of Nuclear Medicine and Radiobiology, Faculty of Medicine and Health Science in the University of Sherbrooke. Dr. Leyton's current scientific impact provides a breakthrough for escaping endosome entrapment, a major overlooked aspect by scientists developing the therapeutics reliant on entering cells for therapeutic efficacy is assumption that the therapeutic was able to freely diffuse in the cytoplasm after internalization. Dr. Leyton's lab was the first to demonstrate the application of cholic acid as a successful endosome escape moiety for biopharmaceuticals. Cholic acid could then be linked to the nuclear localization signals and enhance nuclear localization. Dr. Leyton's lab was first to demonstrate that antibody-drug conjugates could be reprogrammed to localize to the nucleus and enhance their cytotoxic potency. Several international academic institutions integrate ACUM into their therapeutics. So before Dr. Leyton starts his presentation, I just wanted to quickly remind our audience that we will have a Q&A section at the end of the presentation. And if you have any questions during the presentation, you're welcome to post them into the Q&A panel, and Dr. Leyton will get to them after his talk is finished. Now, let's welcome Dr. Jeffrey Leyton. And Dr. Leyton, thank you, and you may begin this seminar whenever you're ready.
Okay, great. So thank you, Dr. Pei and Dr. Zhang for inviting me to present my research to Creative Biolabs, and to your extended community who are also present. Thank you for attending. These are the conflict of interest disclosures before I begin. And so if you're new to the field, or if you're an expert in nuclear targeting, either way, I do hope to educate you today on 3 key learning points And these are why nuclear targeting will improve drug efficacy, how nuclear transport functions, and what are the major challenges for pharmaceuticals targeting the nucleus. So I work out of the University of Sherbrooke, which is located in the lovely city of Sherbrooke, Quebec, which is about 2 hours outside of Montreal and about 30 minutes from the US border. In Sherbrooke is located the University, the Sherbrooke Research Medical Center. It has 4 key infrastructures that I use that are focused on cancer, pharmacology, omics, and medical imaging. And it has a research center that facilitates patient trials. So the first point, nuclear targeting will improve drug efficacy. So I see here that we're like minds, that Creative Biolabs also understands the importance of nuclear targeting. The three modalities of therapy that interest me are, one is gene therapy or vaccine development. That includes CRISPR mRNA transport. The second is radionuclide therapy. And the third is chemotherapy. So the reason you may be asking yourself, why try to specifically focus on targeting the nucleus? Well, the concept is simple, and this is it. The nucleus is the control center of the cell, as it contains the most of the genetic material essential for host survival. Therefore, many in the drug therapy field consider the nucleus a major susceptibility center worth targeting. So interesting to just note that around a quarter of the all approved oncology drugs have their mechanisms of action in the nucleus. So essentially, the concept states that if you can route these drugs more efficiently into the nucleus, you can have an increased therapeutic effectiveness and reduce off-target toxicity, which means you have improved efficacy. So there are 2 major drug delivery vehicles that you can use. One is nanoparticles and one are biopharmaceuticals. I'd like to just use this slide to also clarify that many in the field, in the targeted therapeutic field, as well as Creative Biolabs, utilize cell penetrating peptides, such as the classic one, which is TAT, for targeting the nucleus. It's pretty much well known that if you conjugate TAT to any kind of agent that is specific for a cell surface receptor, it disrupts its targeting ability. And that's because TAT has just a very powerful pull to traverse plasma membranes or any membranes. And so I just want to clarify that my research program focuses specifically on cell surface receptor targeting. And so it employs like a 2-tier targeting system, with receptors as the first tier, followed by the nucleus as the second tier. So these are the 2, in my opinion, the 2 main challenges that have prevented NLS-therapeutics from having a widespread clinical impact. The first is off-target issues that result in dose-limiting toxicities or insufficient tumor killing. And the second is insufficient nuclear localization and hence poor therapeutic efficacy. So I will describe to you my research that addresses in these intracellular barrier, which is endosome entrapment, and also explore nuclear import pathway complexities, and also an extracellular barrier that has to do with cationic charge buildup of your agent. So what is nuclear transport? Well, the story really starts here. And in 1999, the Nobel Prize was given to Dr. Blobel for his discovery of intrinsic signals that govern their transport and localization in the cell. And this is because in 1984, Dr. Blobel and colleagues discovered that this minimal amino acid sequence of PKKKRKV from the simian virus large T antigen and genetically fused to beta galactosidase and needle microinjected, mind you, into mammalian cells was essential for this protein to localize to the nucleus. So hence the first nuclear localization, or NLS, was born. So what are NLSs? NLSs are topogenic series of linear amino acid stretches that are transported into the nucleus. The best described principle is, I would say, is here, which is considered classical, which is called classical nuclear localization. And it starts off here with proteins known as importants, and specifically important alpha, recognize and bind to NLSs contained by proteins in the cytosol that need to get into the nucleus. The second important aspect is the formation of the functional import complex. And now this involves the binding of a second important, which is called important beta 1, and a protein known as RAN, which is either bound in a GTP or GDP or GTP state. And so the third one is the directionality. How does this complex get directed into the nucleus? And this is based off the metabolic GDP-GTP gradient. Essentially, it works like this. RAN is always mostly bound with GTP in the nucleus, and that's due to the presence of a specific protein. And in the cytosol, it's the GTP is hydrolyzed much quicker. And so this starts to build up a gradient between GTP in the cytoplasm and GDP in the nucleus, in the cytoplasm, and GTP in the nucleus. So then this is what drives and gives the complex directionality towards the nucleus. Now I'm skipping over major interactions of how this complex gets in past the nuclear pore complex, but that's not for today's talk. So what are important alphas? Important alphas are basically a kind of a contorted cylindrical shaped protein, kind of like a kidney bean. They have a 30-degree right-handed twist. And importantly, this structural twist provides important alpha with kind of a shallow groove that extends down the middle lengthwise. And at the end of these grooves are what are called the NLS binding pockets. And they're separated into a major and minor NLS binding pocket. And so many years and many significant advances in the field have broken down this table here, which shows that there are now 4 important alpha NLS consensus binding sequences. There's the class 1, which is 4 consecutive basic amino acids. There's the class 2, like the SV40. There's the class, and then there's recent discoveries have now found consensus sequences for the minor binding pocket known as class 3 and class 4. And then there's a bipartite NLS sequence, which is a long NLS sequence that binds both the major and the minor binding pockets. So how is nuclear transport complex? So at this point, I like to just make note that in general terms, this was a great advancement, and the field has started to try and develop nuclear targeting sequences, mostly based on the sequence of SV40, and assuming that this classical nuclear localization mechanism is occurring to their therapeutic. So, but the picture is highly complex for nuclear transport. First of all, for important alphas, there's 7 isoforms of the important alpha. And what's important to know about this is that those NLS consensus sequences, each isoform may bind them differently or not at all. And that's because there's isoforms have various flanking sequence dissimilarities outside of the binding pocket. Also, many of these consensus sequences, they have flanking residues outside, and these flanking residues can make new contacts, or they can change the secondary structure of the actual consensus sequence itself, which will have a different interaction within the binding pockets. Also, certain isoforms are only preferentially present in certain cellular types and states, and that's important for cancer, and I'll get to that in a minute. So to further complicate nuclear transport, there's a completely other side of nuclear transport that is called non-classical nuclear transport. And it's very, very complex. And that's because there's greater than over 20 important betas. And these importance are known as important betas, by the way. They differ greatly in not only in size and shape. As you can see here, these are 3 important betas: important 5, important 11, and important 7. Important 7 will be important to my research, and you'll see that in a minute, but they have various shapes. And so they can bind NLSs anywhere within their central core. And as a result, non-classical NLSs, or consensus sequences for non-classical NLSs, are difficult to identify. So in cancer, many importance are overexpressed and have roles that drive tumorigenesis or progression. And this affects NLS recognition preference and import patterns. So that's something that is very important as well. So hence, this table now summarizes all known consensus sequences, and all I did was add in that there's only been one consensus sequence for the non-classical import, and that's called the PYNLS for the important beta 2. But what does this mean is that there are very few consensus NLSs for use, very little known about nuclear transport biology, specifically its influence in or changes in diseased cells. And therefore, my research program postulates that this is a major reason why NLS therapeutics have yet to make a widespread clinical impact. So that challenged me to kind of really address this situation about understanding more about what are the pathways used in cancer cells. And we developed a proteomic approach to understand nuclear import pathways. And so I'll talk to you a little bit about this, but just before that, again, I wanted to use ADCs, and specifically for one reason is that because we do live in the era of antibodies, and their advantages and limitations provide much room for research and development. For example, unmodified ADCs, antibodies that are kind of reliant on antagonistic mechanisms of action, like blocking receptor-ligand interaction, or for HER2, blocking receptor-receptor interaction, the effects tend to be various and ultimately not curative. On the other hand, ADCs utilize monoclonal antibodies as delivery vehicles. And upon binding to the target surface, receptors, rather than inhibition, target receptors are intended to be portals so that the transported drugs can access the cytoplasm, the intracellular space. So from a targeting perspective, I really liked the ADC targeting concept, and I wanted to combine that with further targeting to the nucleus. And also, in addition, perhaps ADCs can benefit from what my research is going to show you, because one area of resistance is for ADCs is that cancer cells actually keep the ADC entrapped in the endosome rather than delivering it to the lysosome, where the ADC is degraded or pH sensitive linkers release the drug. It's recycled back outside the cell and then rendered ineffective. So ADCs are an ideal candidate to explore and improve the concept of targeting. So the first thing my research addressed was endosome entrapment. And so for this, at this slide, I do introduce to you the Acume technology. And essentially, it's a dual component compound composed of an endosome escape moiety and a nuclear localization. And you can see here that the nuclear localization signal, the NLS that we use is the NLS from SV40. And essentially, I won't go too much into the details on the mechanisms of how cholic acid does cause endosome escape, but it activates the sphingomyelin ceramide pathway, and the increase in ceramide within the inner lining of the endosome causes destabilization and hence lysing of the endosome. My previous publications describe that better. I want to show you kind of how we started the approach of a pending Acume to an ADC, and for the ADC that we chose was TDM1. TDM1 has a DAR of 3.5 and uses a non-cleavable linker called SMCC and attaches to cell surface lysines. So we kind of wanted to follow this model, and we also just decided to attach to lysines, and that's important because as opposed to cysteines, let's say, because lysines, there's a lot of lysines, at least 85, I think, in trastuzumab. And we wanted to generate different species of Acume loading, low, medium, and high. And you'll see what that means later. And so we evaluated different conjugation technologies or methods. And ultimately what we came up with was that SMCC, again, to use to attach Acume, caused the trastuzumab to fall out of solution and aggregate. But if we use the linker with a pegylation in the middle, we eliminated any aggregation at all. So we used then differential scanning fluorimetry to investigate its stability over increasing temperature. And this graph here basically shows you that the unfolding pattern doesn't occur until well after 70 degrees Celsius. And that indicates that the Acume modification of TDM1 is stable at temperatures compatible with human use. This one shows you it's a kind of a modified saturated binding curve, but we also are evaluating internalization. And this shows that that Acume modified TDM1 had comparable rates of HER2 association and internalization to TDM1. Hence, it didn't affect the binding. And that's an important point. So what we see here is now how does the ability of Acume, can it localize an antibody to the nucleus? And so the top figure is our initial work in leukemia cells. And so this antibody is specific for a receptor called CD123. And the figure demonstrates right here that Acume modified antibody accumulates considerably more in the nucleus. In contrast, unmodified antibody or NLS modified antibody modified with only NLS remains trapped inside these endosomes. On the other hand, if we were to modify antibodies only with cholic acid or take Acume and modify the NLS signal sequence, well, we get endosome escape, but no nuclear localization. The bottom figure now is with TDM1. And this model system is GIMT1 cells, which are HER2 positive, TDM1 resistant. And they were known, previously published, they were resistant because of rapid recycling, as I mentioned. So this shows you that basically, if we follow trastuzumab will always stay kind of near the cell surface. We stained recycling endosomes using RAB11, which are indicative of recycling endosomes, and you see that they're abundant around the cell surface and in the cytosol. And then if you overlap them, you see that there's a co-localization within the cell surface, indicating that yes, their TDM1 is most likely being recycled back outside the cell. We also stain for ceramide, which is the indicator of endosomal membrane disruption. And as we would expect, we don't see any ceramide with TDM1 treated GIMT1 cells. But when we treat with acute TDM1, we see an abundance of green. And we see that this green is co-localized strongly with RAB11 endosomes. And essentially, this indicates that acute TDM1 is undergoing its mechanism of endosome escape. We fractionated, we did cell fractionation to obtain a pure and intact nucleus, which we then lysed and ran on an SDS page and then probed for the antibody portion of the ADC. And as expected, we don't see anything really of TDM1 in the nucleus, but we see an abundance of accumulated acute TDM1 in the nucleus. So now, does this mechanism result in any improved efficacy? Well, it does. So the left cytotoxicity assay, and we're using SKBR3 cells here, acute modified TDM1 increases cytotoxicity up to 18-fold. We tested, again, low, medium, and high loads. Just a bit of a word on terminology on that. It's that we're unable to actually quantify the number of peptides. So we separated them into how they migrate on an SDS page. And so we would see that low modifications migrated a certain distance, and the higher loading, the higher on the gel they migrated. So we used those 3 different species for that. I can explain that later if you would like. Importantly, acute modification does not abrogate HER2's target specificity. So with MCF7 that are kind of considered HER2 negative cells, we don't see any cytotoxic effect, and we're using the highest load. Importantly, acute modification alone on trastuzumab does not cause cytotoxicity, and that's an important feature. And lastly, if we just modify TDM1 with NLS only, we see significantly weaker cytotoxicity. So those are kind of like the effects of what we saw. We wanted to explore now like how this is happening again, and we wanted to really, this started with just to try and confirm that important alpha was taking the acute modified TDM1 into the nucleus. So to do this, though, we had to set up a proteomic approach. But first, we had to set up a kind of a biochemical bait and prey approach. And this slide just sums up years of hard work from the master student who did this. And I'll just kind of show you like what we did and the challenges that we overcame. So one was, it's about sensitivity, getting enough pull down of potential interactors to inject into the mass spectrometer to get some workable data. And that's really reliant on this upstream process. Long story short is that we had to use kind of really high concentrations of ADC. And we tested various amounts of cells, and we had to finally found that we had to use 15 million cells, which is a lot. Next, we kind of had to confirm that we wanted to do the capture of interactors in the cytoplasm. We wanted to maximize the amount of acute TDM1 in the cytoplasm post endosome escape. And so for that, we kind of had to do some kinetics studies. So one thing here is that we saw is that by 2 hours, we saw that internalization was completely done. And so we knew then at least to not stop the reaction and lyse cells before 2 hours. Second of all, we were using high amounts of ADC. We were worried that actually we would be killing the cells and we would be harvesting dead cells, and this would complicate our approach. So what's interesting enough is and we couldn't do kind of a standard Presto Blue or cytotoxicity assay that measures kind of mitochondrial redox levels that's traditionally used, and you read those assays out to 72 out to 96 hours. We needed to do this within the 2 hours. So for this, we looked at DNA integrity, just as a simple way to kind of look if there was any DNA damage. So we could do that using propyridium iodine staining. And what you see here is that actually TDM1 caused a little bit of 15% of killing or of DNA damage within this 2 hour window. We had to live with that. But what's nice is that the Acume, in this case, the Acume medium loaded TDM1 really didn't cause any DNA damage. And that's nice to know. We can talk about that further if you like, but we were very happy with letting the incubation go for 2 hours, lysing cells, doing a pull down with protein G. We had to modify the type of protein G coated beads that we used. And essentially then it came down to these types of SDS page gels that we used as our final readout. And so what this shows you is that like Laymoon 1 is we just spiked an SDS well with 5 micrograms of TDM1 and just went and saw, okay, this is what the band looks like just with 5 micrograms of TDM1. So we treated cells with trastuzumab as well, and we did the same process, and we pulled it down, and we were able to get nice signals of pull down trastuzumab. This is just what the lane 3 looks like with no pull down. Beads only, we see blank because we're not pulling down any antibodies. But here came the real challenge. So if you just go to the far right and you just look at TDM1, TDM1 was difficult to pull down. As you can see, the signal is much weaker than just trastuzumab, and that's most likely due to modifications of attaching TDM1 to the lysines. They interfere with protein G binding. And so sure enough, what we saw here is that if we tried to pull down an Acume high modified TDM1, we weren't able to pull it down. And barely able to pull down any Acume medium loaded TDM1. And we were able to pull down sufficiently amounts of Acume low TDM1, albeit much lesser than TDM1, but it was good. It worked for us. And because we were able to get enough signal from the mass spectrometry machine. So moving to the proteomics method of how we did this, we did 2 kind of different and complementary systems. First of all, we were able to pull down around 1,700 unique protein groups identified by mass spectrometry of the co-immunoprecipitated proteins. And then these interactors were fed into a software called Significance Analysis of Interactome, or SAINT. And SAINT is basically a software for scoring protein-protein interactions based on label-free quantitative proteomic data, example, spectral counts or intensity values. However, we use to our advantage that there's really no kind of one size fits all for statistical modeling for these data sets. And that's because the Saint software is very sensitive to your experimental question or design, specifically meaning to the amount of baits that you use versus the amount of controls that you use. So what that means is that we were able to kind of set up two systems. We set up one, what we call the one bait three control, or the 1-3 system, where we use as our experimental data set the Acume low TDM1 only, and what are the interactors that were pulled down. And as controls, we use TDM1, trastuzumab, and beads only. So TDM1 is to kind of eliminate any false positives that are interactors that may be stuck onto the drug. And the TMAB is to remove any interactors that stuck onto the actual trastuzumab backbone. And then we did an analysis where we set up another system called the 3-bait 1 control, or 1-1-1-1 system. And that's where we treat each bait as an experimental group, and we only have one control group. Very briefly is that around 18, so in the 1-1-1 system, we can set up a Venn diagram about what were the unique interactors for Acume TDM1. And just briefly to show you that approximately 18% or 206 were specific for Acume TDM1. So what the Saint system allows you to do is to control the stringency on what you identify as a specific interactor. Specifically, the scoring is done on a scale of 0 to 1. 1 meaning that you have 100% confidence that two proteins interact together. And so we set up a scale where we found like a sweet spot, basically, where we only collected interactors who had a score of 0.9 in the 1-1-1 model, and we set up a score of 0.5 for the 1-3 model. And then what we did is we plotted the 1-1-1 model over the 1-3 model in order to get an overlap or sweet spot of what are the actual importance that were interacting with Acume. And that's how we did it. And so the data revealed this. So quantitative data, what's called a bubble plot, which is kind of the Saint score, and versus the spectral count, or the overall abundance or presence of the important alpha. And if you look at the data like in this fashion, you see something striking. You see that the highest present, although not the highest Saint score, but the most abundantly present was actually IPO7, and IPO7 is important 7, a non-classical important. We do see important, the one that we were expecting, important alpha, which is the orange circle here. But if you look at it from a different point of view, which is called a quantitative dot plot here, where you can kind of compare Acume TDM1 to TMAB and TM1, you see that IPO7 again, important 7 again, is really the strongest interactor. So we couldn't believe this at first, and we wanted to see this biochemically. And sure enough, if we pull down the important interactors and we probe the pull down interactors on a SDS page and do a Western blot probing a specific importance, you see that yes, Acume TDM1 did pull down important alpha, but nowhere near as strong as it pulled down important 7, and that was like a very very exciting kind of result. So the question is, what does that mean in terms of nuclear transport and efficacy? So what that means in nuclear transport here on the right is that you can see here is that we knocked down selected importance thanks to the proteomics portion, because there's so many importance. We cannot just do knockdowns of all of them. The proteomic approach was vital to actually narrow down the collection, and then we can target those importance, and we knock those down by sRNA. And so what you see here is that basically uh, in the red, if tracking the trastuzumab portion of in cells treated with acute low TDM1, you see nuclear localization here, but it's knocked out. There's no nuclear localization in cells that have importance 7 knocked down. And that was like a really striking feature. Just to show you how kind of it works is also, you can see the levels of importance 7 are knocked down tremendously in these cells versus the siRNA controls. And in TDM1, we did that as control, but we weren't expecting anything anyways. So what does that mean for cytotoxicity? Well, it means this. It means that it was really only when importance 7 was knocked down did we lose a significant amount of cytotoxicity potency with the acute modified TDM1. And this result really demonstrated a lot. And so the kind of mini conclusions here on what the significance of this work was is that this demonstrated, first of all, that Acume technology is able to link endosome escape with increased nuclear localization, and it improves cytotoxic efficacy, at least for TDM1. This was a first demonstration of a methodology that can successfully not only interrogate, but determine precisely the required nuclear transport pathway for an NLS therapeutic. And the NLS therapeutic nuclear transport pathway cannot be assumed based on the NLS sequence you're using. Granted, there are limitations. Thus far, we've only done this for a single cell line. The methodology is right now only optimized for kind of antibody pull downs, but I'm sure that we could also do with other nanoparticle things, as long as there's a pull down capture system. And most importantly, I think to be fair to science is that it's unknown whether the importance 7 or the classical important alpha, important beta 1 was the naturally dominant nuclear transport pathway for this cell type. That's a key point. Like we don't really know. Maybe importance 7 was actually the preferred pathway for some reason in SKBR3 cells. We don't think so, but again, thinking that it's very easy to fall into that trap of assuming that the classical nuclear transport pathway is the one that's always occurring. But that's just to be fair, I wanted to mention these limitations. So moving on to kind of how do we overcome the barriers based on cationic charge, and I'll explain to you a little bit about that. And they touch both intracellular and extracellular barriers. So the intracellular part is this. So you may be asking yourself why you thought importance 7. Well, if you look at the crystal structure of importance 7, it has a cavity right here that's lined with acidic amino acids. But more importantly, it has this long 90 amino acid acidic loop. And it's kind of opens and shuts like a lid. So I think it's very tempting to kind of think that that this may be occurring, that the importance 7 is basically capturing a Q modified TDM1 because the SV40 NLS has a net cationic charge of +5. And now because we do chemical coupling and the field does it this way, you're adding in different loads. So you're adding multiple NLSs to your therapeutic target agent. And this, we surmise that basically the working model is that a CUM or any cationic NLS modification results in kind of a supercharging of the overall NLS therapeutic, and hence important 7 may be the dominant nuclear transport. And that's kind of the working model there. Now, cationic charge-based barriers for NLS therapeutics and the extracellular challenge. And this is kind of a bit more known. There's great reviews from Wu and Center and Sui et al. At J control release. And that works like this. Essentially, agents that are injected intravenously get out of the bloodstream and kind of traverse the tumor environment, sampling the tumor cells, getting internalized, and ultimately having a high uptake. I just mentioned that, while NLS's modification of nanoparticles or proteins can cause this supercharging effect. Well, it's kind of known that with like supercharged agents, they get trapped in what's called the reticuloendothelial system, and they are kind of hallmarked by kind of like a more negative net charge membrane, cellular membrane. And what this means is that if you, for a typical example, if you inject just an unmodified antibody, the solid line red here means that it gets eliminated slowly from the blood, and that results in a high tumor uptake out to a few days. But for kind of a supercharged agent, the sequestration in the RES will rapidly eliminate the blood, and this negatively impacts the amount of tumor uptake. And this causes poor therapeutic efficacy, can cause poor therapeutic efficacy. So lastly, I'd like to just kind of talk about the work that we've done on this. I used an antibody specific for a cell surface target called CD125 that is overexpressed in bladder tumor models. I was fortunate enough to work in the Molecular Imaging Center at Sherbrooke, where we use PET imaging technology. I don't have time to go into it, but we use copper-64 as our radioisotope. And essentially what you see here is that this mouse with the high expressing and the low expressing CD125 positive tumors, what you get out of this is that the acute modified antibody has strong tumor uptake relative to the unmodified antibody, or an antibody, the antibody just modified with NLS only. And this is at 24 hours and 48 hours post-injection. But we do see sequestration within the kidneys, specifically the glomeruli, with the acute modified A14. And it did reduce by 48 hours and decrease to background levels by 96 hours. But nonetheless, we did see this sequestration, which indicates that there is some sequestration within the RES. For the NLS, you don't see it. And that's just because we don't have the PK curves to show you, but it just really just crashed out of the bloodstream. And really, you can see that it barely even lights up the tumor. It can't even pick up, it doesn't target at all the lower expressing tumor. So this really shows you why, and the big challenge with NLS therapeutics. So my last part of the talk, I am running out of time, so I'm going to try to finish in the next few minutes. We set up a kind of scientific flow to generate zero net charge NLSs with the hypothesis that if we eliminate any cationic net charge and get that to zero, we can overcome this several of these problems that I showed you. So we went into the we retrieved crystal structures from proteins involved in virus infectivity, transcription initiation, and RNA processing. And we did through modeling, essentially to identify the sensitive and non-sensitive residues we could manipulate. The non-sensitive residues that did not affect binding, we then did residue scans, all with the aim of inserting residues for achieving a net neutral charge. And then we did through various statistical algorithms to really be confident that the binding is either improved or comparable to their parental NLSs. So just to show you kind of what this did, a unique group that we're working on are NLSs that target the minor binding pocket. And just to show you kind of what is happening is, like in the wild type NLS, there's no interaction, but with our new NLSs, and in this case, we're inserting an acidic residue kind of to bring down the charge to zero, we're establishing novel contacts. So we're working this up and what it means scientifically, just to identify kind of new NLSs in nature, nothing to do with therapeutics. And you may be asking, well, do they really work? And so this is ongoing, but just to show you, the answer is yes. Many of them work very well in mammalian cells. So the research conclusions here is that it's feasible to stably functionalize TDM1 with a CUME. CUME enables endosome escape with TDM1 nuclear targeting and cytotoxicity, which improves it. We developed, I showed to you, a novel proteomic approach to interrogate and accurately determine dominant nuclear transport pathways. I also showed you how cationic charge is both an intracellular and extracellular barrier, limiting therapeutic targeting, or at least modifying therapeutic targeting to the nucleus. Novel zero net charge and functional NLSs have been generated. So the ongoing research avenues are the following. Specifically for the ADC people, we're now matching payloads that are active in the nucleus with a CUME. We're determining the nuclear transport pathways in various HER2 tumor cell lines. We're ranking the zero net charge NLSs, ultimately to kind of substitute the SV40 NLS with our novel NLSs and test with ADCs or any targeted therapeutics you may have if you want to collaborate. And lastly, the modified or enriched overarching learning points, I think for the audience, I hope they appreciate it, is that drug therapeutic scientists gained an overall insight to biopharmaceutical design with nuclear targeting applied to ADCs. You also gain specific insights on the importance of understanding nuclear transport biological complexities. You are provided with hopefully a guide from a biological viewpoint of how you can use that in parallel when designing your own NLS therapeutic strategies. And for the general audience, nuclear targeting is of high interest, but there are major challenges and innovative advancements were learned for mitigating these challenges. So I'd like to just acknowledge several of undergraduates, graduates, postdocs that I was privileged to train and who contributed, but specifically Dr. Simone Baudouin and the University Peptide Synthesis Platform, who makes any NLS that you need. Also, the University Proteomic Platform, specifically master student who got his master's in my lab, Vincent Lacasse. And this could not have been done without actual proteomic expertise. And that is from Dr. Steve John. The PET imaging portion, the work was really done by Dr. Michelle Paquette, and we thank the Sherbrooke Molecular Imaging Center personnel and also personnel at the University Hospital Radiochemistry Service. And lastly, the zero net charge NLS portion, I had a real great pleasure because this was done mostly by undergraduates using the university research training program there. So I had a real privilege to work with them. And then these are the people who provided not only money, but also like infrastructures at the university for allowing me to realize and present this work for you today. So thank you. I'll leave it there. If you want to contact me, here's my email address.
Right, thank you, Dr. Leyton. Great presentation. And I'm sure our audience enjoyed this presentation as well. And to all the audience, you can access the recording of this webinar at our official website in 2 working days. So, okay, let's get into our Q&A section. First question is, could you modify the regular IgM or IgE antibodies for the infectious disease?
Well, that's a great question. And that's why I really wanted to, I was very happy to be invited here, especially with the pandemic. It's difficult to kind of disseminate this work verbally, orally to the people there. I imagine there's a way to do it. I would have to kind of hopefully speak with, an infectious disease expert to strategize on how to do this. So I think so, but I don't have any kind of definitive answer for you.
Gotcha. Okay, the second question is, do you have any experience using your antibodies to gain access inside the platelets? I mean the uncolored blood cells.
It really just, all we need is a target, a cell surface target. So if you have a good target for any cell type, we can theoretically do this.
All right, the next question is, since you mentioned the antibody modification with cholic acid coupled NLS can increase the antibody intracellular accumulation in target cells. So the question is there a recommended ratio of NLS to the antibody?
So this is maybe where the ADC community can really kind of help any NLS modification of therapeutics. It's because we did try to kind of do the traditional like DAR for this, and it's very, very difficult. We haven't been able to do it through mass spec. And just so for your ADC audience, if you notice, sacituzumab govitecan from that was originally started from the Goldenberg group and Immunomedics. They're the ones who kind of made a DAR 8 of SN38, and they were able to do that because of their PEG hydrophilic linker. And if you go and read all their literature, they never report a mass spec derived DAR. It's extremely difficult. They use kind of very unique ways through multi-dimensional liquid chromatography. So the answer there is that we just only have approximations of the NLS, unfortunately. It's not a problem in terms of like injecting or for use. It's just, yeah, quantification on this. We are trying to work with kind of more spectrometry ways to look at this, but it is a difficult area. Yes.
Got you. So we have limited time, and the last question is, could you please give some future aspects for the NLS-therapeutics in the antibody drug development regarding their like delivery efficiency, the stability, or the potency?
Right. So my research program will focus on really matching kind of ADCs with with nuclear active payloads. And in parallel, my science is also kind of developing this new novel NLS sequences, specifically with a zero net charge. I'd like to then kind of combine those two and to study pharmacokinetics, tumor targeting, using the power of molecular imaging, which is I'm lucky enough to be at the university that has this. So that's the plan for there, and essentially then test various ratios of modification, DAR also, and see which have the strongest anti-tumor killing, and see how we can advance that into kind of phase 1 clinical trials.
Nice, nice. Thank you. So it's time to write it up. And please note that Creative Biolabs regularly invite brilliant experts in the fields of life science and biotechnology to present their most recent findings in our webinar series. And please visit our website often and subscribe our social media so that you won't miss our upcoming webinars. So that's all for today. And thank you everyone for joining us. And thank you, Leyton, Dr. Leyton. And we will see you next time. Thank you.